So now that we've been introduced to three major types of DNA sequencing methods, it's now time to talk about the dilemma we left off with, which is how do you get a whole lot of DNA if you need it? So in this video, I'm going to talk about two techniques, gene cloning and the polymerase chain reaction, both of which are ways that you can potentially get a whole lot of DNA. The original method out of these two that was used was gene cloning, which involves creating recombinant bacteria. And later on, once we had a better understanding of the process of DNA replication, then we're able to basically create conditions to artificially replicate DNA using PCR. So we'll start out a little bit about gene cloning in E. coli. I talked a little bit about this previously, but in addition to having a single circular chromosome, bacteria also have separate pieces of DNA called plasmids. And E. coli can actually exchange these plasmids with each other in a process known as bacterial conjugation. So when we figured out that bacteria had these plasmids, that gave us an idea. We could actually make artificial plasmids and get bacteria to pick up those plasmids, using this process of transformation. And so then, if we make an artificial plasmid, here's some elements that could be really useful to add into those plasmids. So you probably want a plasmid that contains the gene of interest. Along with that, you can also add in some screening genes. So for example, a gene that codes for a protein that makes bacteria resistant to antibiotics, or maybe a gene that turns that bacteria fluorescent, for example. And then if you're using a eukaryotic gene, you also need to include some kind of expression vector. So that's a promoter sequence that the bacterial RNA polymerase will recognize. So if you put all these things together, stick a plasmid into an E. coli, as that E. coli develops and replicates itself, it will replicate all the DNA that's in that plasmid, and it can also share that with other E. coli, so that pretty soon, if you allow enough generations to pass in a vat of E. coli, you'll wind up having a bunch of copies of the piece of DNA that is of interest. And there's other applications for this method also. So, it could be useful for generating extra DNA, but it could also be useful for generating extra protein. And so this is something that as soon as we figured out how to do it, one of the first things we applied that to was for the production of insulin by E. coli. So originally, for someone who's diabetic who needed insulin, we'd actually harvest the insulin from pigs, and it would take a lot of pig pancreas to get enough insulin to treat just one person with diabetes. Once we figured out how to engage in gene cloning, we could then get E. coli to express the insulin gene and produce insulin protein and purify it from the E. coli and make way more insulin for the amount of resources and effort involved. And so that's what's really allowed diabetic people to survive, is that process of gene cloning. That's not the limit. Similar methods can also be used to amplify a gene, get extra copies, and then insert those extra copies into other organisms. So for example, you could insert genes that make plants more resistant to pests. So one of the early examples of that was using a toxin from bacteria, the Bt toxin, which is now very commonly inserted into different plant genomes to confer resistance to insects. This has also been a method used to alter bacteria to allow them to metabolize different types of waste, particularly waste from oil spills. And in addition to insulin production, same method gets used to make different types of growth hormones for people that aren't able to produce that themselves, and other proteins for other functions. So the example here is a protein that will dissolve blood clots when someone has a heart attack. And I also just wanted to note that in addition to using E. coli for this purpose, yeast have also become an important type of cloning organism, very widely used as well. But that might lead to another question, which is how do those recombinant plasmids actually get made? So that got started with the discovery that a lot of bacteria contain a type of protein referred to as a restriction enzyme, which is a protein that can recognize a specific sequence in DNA and then chop DNA at that specific sequence. So here it's the sequence up top, GGA-TCC, and you'll notice that it is symmetrical across the complementary sequence. So in the opposite direction, that same sequence GGA-TCC. And so when a restriction enzyme, in this case it's one that has the name BAMH1, encounters this sequence, it will chop it and it will leave free nucleotides at either end. So we refer to those as sticky ends. So what this means is you can use restriction enzymes to chop up a bunch of DNA at known locations. And then if you create those ends at either end of something else that you're interested in, then you can introduce those into a plasmid and then seal up those gaps with some DNA ligase, and then you wind up with the recombinant plasmid that you're interested in. Okay, so that's one way to generate a lot of DNA, and it was the first method that was used, but a much more commonly used method now is a method referred to as a polymerase chain reaction. And so this has the benefit of allowing you to amplify DNA without needing to use bacteria, and it takes advantage of something that can happen when you heat and cool up DNA. And it is much, much faster and cheaper than using recombinant bacteria, because you can generate billions of copies of DNA in just a few hours' time. So the ingredients for the polymerase chain reaction are very similar to what I showed you for Sanger sequencing, right? So you need whatever DNA you have that's of interest. Then you also need, again, sets of DNA primers. And in this case, you need two primers, a forward primer and a reverse primer, and I'll talk a little bit more about what that means in a moment. You need your nucleotides, so that was your dNTPs, and then you need a DNA polymerase. Okay? So here in a nutshell is kind of how this whole process works. So if you have a double-stranded piece of DNA, so here's your original DNA sequence that you're interested in, and if you heat DNA up, the two strands, the hydrogen bonds holding the two strands together, will come apart first as you heat up a piece of DNA. So if you heat it up, that will denature it. And then if you let it cool down again, if you have a whole bunch of primers present in your mix, DNA primers that match up with the sequence of this DNA, as you cool this back down, what can happen is one of those primers may wind up binding along or somewhere along your piece of DNA, flanking the region of interest. And again, there will be, for any given sequence that you're interested in amplifying, there's going to be a forward primer and a reverse primer, so a pair of primer types that are in this soup. Okay, and so here here's the 3' end of the original DNA. Here's the 5' end of this primer. Here's the 3' end. So if you cool this down, if you've also got dNTPs and DNA polymerase in the mix, then that DNA polymerase is going to elongate each of these pieces of DNA. And it's going to keep going for, some extended length here. And same thing true on this other strand on the other side. Let me erase some of this other stuff so it's out of the way. Okay. All right, so it's just going to keep elongating for however long you leave it at that cooler temperature, until you reach a point where you heat this mixture up again and you repeat the process of pulling apart the two strands and denaturing them. So if we do that again here, let's just take a look at what we've got. If we do that, So I'm just going to move this piece, or the original DNA, over. All right, so here's my original piece of DNA. Here's my other original piece of DNA. Okay, if we cool this down again, now I'll have another one of these red primers bind on this leftmost strand. And I'll also have red primer that's going to bind on this green strand in the same position, right? Because this leftmost piece of DNA has the same sequence at that position as this green piece of DNA does, okay? Hopefully you can see that. And that might also suggest to you, okay, so then if we look at this blue primer, you know, okay, we'll have that blue primer bind to that other piece of DNA, the complementary strand from the original one. And then also that blue primer will be found at that same position on this green piece of DNA. And so it's actually these green pieces of DNA are the ones to really watch through this next process. So now we've got that primer has bound, or we call that the annealing stage. And so now it's possible for DNA polymerase to come in and do its job. So it again is going to elongate in the 5' to 3' direction. So on this green piece of DNA, it's going to elongate up until it hits the end, right, where that other DNA primer was. Okay, so that piece has been replicated. Elongation on the other green piece is going to travel in the other direction. Okay, so we've got elongation there. And the same thing's going to happen on these original pieces of DNA as well, as previously. And those are going to generate segments of various unknown lengths. Okay? But now, if we look at this again, if we go through this process one more time, we're going to start winding up with pieces of DNA that are all the same length and that are bordered by the 2 primers that were put into the mix. So this method over time is going to allow us to amplify a given stretch of DNA that is bracketed by those primers. And this is an exponential process, right? So from round 1 to round 2, I went from 1 piece of DNA to 2 pieces of DNA. Round 2 to round 3, I went from 2 to 4. Then from there, I'm going to go from 4 to 8, and so forth and so on. So over a relatively short period of time, you can go from just one section of DNA to many, 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 many copies of that piece of DNA. And it's a very precise process. And if you have a fairly long primer, it can be specific to just one location in a genome, so you can amplify just one gene at a time. If you have shorter primers, then you're going to probably wind up amplifying a lot of different regions of DNA simultaneously. So you can amplify an entire genome if you have a lot of very short primers. But there's a slight problem here, which is this process is dependent on action of a protein, DNA polymerase. And as I hope I've emphasized up until this point, proteins are very sensitive to changes in temperature. They're also sensitive to changes in pH. And this process that I've described, the process of pulling the 2 strands apart, denaturation, requires heating the DNA. So that could cause this DNA polymerase to also denature, and then be non-functional. So originally, when biologists encountered this quandary, basically what they do is every time you reach the extension step, they would dump in more DNA polymerase. But DNA polymerase gets really expensive. So again, we're limited in how useful this process was. Thankfully, though, we discovered an organism that is actually very heat tolerant, that has a special DNA polymerase that is also heat tolerant. So it evolved in hot springs in Yellowstone. It's a bacteria called Thermus aquaticus because it lives in these thermal pools in Yellowstone. And so this is a heat tolerant form of DNA polymerase. So I have another question here on the slide, which is, how do you ensure that you get only one specific gene region? And hopefully I've shown you or talked a little bit about the answer to that question as well, right? Design primers that are unique to that particular region in the genome. And again, you have to use both a forward and a reverse primer to really bookend the area that you're interested in. Okay, so that takes us through techniques that let us generate a whole lot of DNA. And so now we're going to shift topics a bit to talk about methods that are used to study more downstream processes, so studying gene expression patterns. And so in the next video, I'll talk about in situ hybridization and the technique of RT-PCR.